Good
Done,
afternoon
ma'am.
to one and all present here for today's session. Today we are going to have a wonderful session on how to write research papers. As all of us know, we are moving towards the road which is called as research, where research includes literature review, wet lab, dry lab, and after these three important steps, the next important step is to write a research paper for publications. So, though it is one part of your research, many a times students, scholars, they get stuck up in the process of writing the papers. So, today we are having a very wonderful guest speaker, Dr. Aparna B. Gunjal, and we are going to have a session on how to write research paper. Before we start the session, I would like to welcome Dr. Aparna ma'am on behalf of Microbiology Society. We welcome you, ma'am, for today's session. Let me introduce our today's guest. Dr. Aparna ma'am has completed her graduation from Anasaheb Magar Mahavidyalay Hadapsar
But as you
MSc
know, sorry.
from I request the audience to please mute. Sorry for the interruption. Dr. Aparna ma'am has completed her graduation from Anasaheb Magar Mahavidyalay Hadapsar, her post graduation from Modern College Arts, Commerce and Science College, Ganesh Khand, and she has completed her doctorate in environmental sciences, subject from Savitribai Phule Pune University, Maharashtra. Currently, she is working as assistant professor in Department of Microbiology at Dr. D.Y. Patil Arts, Commerce and Science College, Pimpri, Pune. Her area of research or her research areas of expertise are solid waste management, plant growth promoting rhizobacteria, e-waste management, bioremediations, etc. Aparna ma'am has 190 publications to her credit. She has 15 national and international awards to her credit. She has worked on composting aspect as a senior research assistant at Hong Kong Baptist University. Aparna ma'am is also an editor and reviewer for many journals. So today we are having a very wonderful speaker who is going to guide us on how to write the research papers, and it is our privilege to have her for today's session. Aparna ma'am, please take over the session.
Yes, thank you for the nice introduction. So, the today's session is
What is metabolism? People say there's
how
somebody
to
is so
write
metabolic, somebody.
the good research papers, okay? Which is there. So, I just want to know. I hope the slides are visible just before I can start.
No, ma'am, again, it's not visible.
Yes, let me know once it is visible.
Yeah, yeah. Now it's visible. It's coming and going, ma'am. It's not
No,
very
no,
steady.
I'm just no, it's not coming and going. I'm just like, can you see the title now? Research article and its structure.
No, 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 ma'am. The screen is only not visible.
Okay, just a minute. Yes, now.
No, ma'am, still it's not visible. Some problem, technical problem from your end. Now it's visible, ma'am.
Now it is visible.
Yeah,
Yes.
yeah. But the slides are not visible. The screen is visible.
Okay. Uh, just a minute. I will just try from my another this thing. Just give me a moment. I will have to try from my another desktop. Just
Sure,
give me a
sure.
moment. Yeah.
Yeah.
Megha, now you are in Mysore or Bangalore?
Bangalore, sir.
When you come by Monday, Monday
Sir?
to Friday. When you visit
Hello?
Mysore, I said.
No, sir, actually, I'm located in Bangalore itself, but I'll visit to my guide once, like almost 15 days once.
Oh,
By the time the guest speaker is available, can I please have the details? Like Dr. Ambika ma'am, can you please introduce yourself?
you are doing PhD part time.
Yes, sir. Yes, sir.
Good afternoon, everyone. I'm Dr. Amrita Nayak. I'm pursuing my PhD degree at Indian Veterinary Research Institute, Bareilly.
Okay. Thank you. Dr.
Welcome, ma'am.
Majid, sir.
Hello, am I
Hello.
audible? Yeah.
Yes, sir. Yes, sir. You're
Good
audible.
morning, everybody. This is Dr. Majid Mohiuddin. I'm from Hyderabad. So I have done my PhD from environmental science and technology, and my major area of research is microbiology.
Okay. Okay, sir. Thank you.
Thank you.
If any other new participants are there, they can introduce.
Yes, sir. Yes, sir. The participants can introduce themselves by the time our guest speaker is available.
I see 51 participants today. It looks a good
Yes.
number.
Yes, sir. Yes, sir. It's very good to see all of them.
Exactly. Actually, I will log out because I am in a train
Okay.
and I will reach Mysore in a few minutes from now.
Okay. Okay, sir.
Today is good number.
Yes, sir. It's very nice to see these many participants today. And still people are joining for the session.
Yeah.
Because topic is very interesting, so that's why everybody is curious, I think.
Yes, sir.
Patel sir, namaste.
Namaskar.
I guess Sanjeev sir,
Namaskar.
Prakash sir has some
Karalur,
connectivity
sir,
problem.
Kopargaon वापस जाता हूँ.
अच्छा. You are going.
No, I don't have connectivity problem. I am in train.
Yeah.
कोपरगांव से वापस जा रहा हूँ.
हाँ.
Today,
Aparna ma'am, can you please check the screen availability? Yeah, your screen is visible, ma'am.
Yes. So is it visible now? I hope now it is okay.
The screen is visible, but the PPT slide is not visible yet.
Yeah, now.
No.
But I have done the sharing. So please, you can ask some other participants also if it is visible.
No, it is not visible.
Not visible.
We can't see anything actually. Dr. Aparna, you can take someone's help actually.
No, I have done sharing. So, one moment.
आपण काहीच होत नाही.
Now it's visible, ma'am.
it's visible, right?
Yeah, now it's visible.
Okay. So
Thank you.
Yeah. So can we proceed?
Yes, sir. Yes, ma'am. Yes, please.
Just a moment.
Ma'am, you can start.
Yes, 1 minute. I'm just checking the sound connection. Just. Yeah. So sorry for the delay. Now a research article, like a research paper. We have the research paper. I hope I'm audible. Yes.
Yeah, you're audible, ma'am. Please make
Perfect.
it full screen.
Yes. Is it full screen?
No.
Not yet, ma'am.
But it is visible.
Yeah.
Yeah, it's visible. You can carry on.
Yeah,
You
yeah.
can say.
So, in a research paper, we have we are following globally the IMRAD pattern. So it is introduction, materials, methods, results, and discussion. So that is the standard pattern that we are following globally, which means over the world we are following this pattern, and this is the simplest pattern and the most standard pattern that we are following for writing any research article. Okay. Now, the first thing that you need to have is the title. Okay, so your title, which is there of the research article, it has to be very precise, short, and clear. The meaning is that, okay, because lengthy titles, they are not good enough for the good quality of a research paper. So the title has to be very short and clear. Because lengthy titles, they are not accepted even by the journals. So for that need to see that the title is very sweet. In some of the journals, after the title they ask subtitle. Now the subtitle, it says that whatever the title you have selected, in that only still more you have to shorten it and see to it that you give some subtitle for your research article. Okay, so that is called where your important keywords will be there for the subtitle. Then comes the name of the authors, or you can say the author. In some cases, the thing is that you only one single person is there, okay, who has carried out the whole research work, or in some cases there is a team of people who is where they have carried out the research work. So in that case it becomes a team, and there can be more than one author, or in some cases there can be, as I mentioned, only a single author. So X, Y, and Z. Here I have just randomly taken where there are different authors. So X, Y, and Z. So you can say that it is a team, okay, where there is not a single person who has carried out the research work. And the same thing it is shown where you can see X1, Y1, and Z2. A star is given. Now, one which is there, it stands for the affiliation. So after the name of the authors, you have the affiliation of the authors. Affiliation is nothing but it is the institute, the name of the institute. If you see the first example, it says that it is X1, Y1, Z1 star. It says, or it conveys, that all these three authors which are there, or this group which is there, is from the same institute. Or it says that the name of the institute, they belong to the same institute. In other words, that is, we can say. And star which is there, which you can see, a very small star, it says that the person is the corresponding author. In a team, we require a corresponding author. So here, the Z which is there, you can see there the star is given. So it says that that person is the corresponding author. The corresponding author, the meaning is that whatever, because after you have written a research paper, it has to be sent to the journal for publication. Now, in the journal, you have the editor, so editorial team. So that editorial team which is there, they will convey whatever the result is there of your research paper. There are three criteria which can be there, okay, three options. Either your research paper is they will say that it is minor revision, or they can say that there is major revision, or the third option is the rejection. So any three possibilities can be there. And this result which is there, it is communicated to the corresponding author, because corresponding author is responsible for all the things. Whatever the communication is there from the journal, that will come to the corresponding author. And the corresponding author has to convey that message to the remaining co-authors which are there. Okay, so that is how it follows, and hence the corresponding author is important. The email ID of the corresponding author is given. Okay, it need not be that you mention the email ID or the email address of all the authors. Okay, because corresponding author will be handling all the communications which is there from the journals with respect to the paper. The other option is what you can see is X1, Y1, Z2 star. Here it says that 2 which is there. It says that the last person who is there is from different institute. Whereas X1, Y1, because it is 1, 1. So it denotes that they are from the same institute. Okay. So if the institute is different, you have to denote it by different letters or by different alphabets. Okay. Numbers you can say, or by different what you can say, the letters also. Okay, alphabets will also do. So here, as I have shown, X1, Y1, Z2. Can you see the next slide? Yes. Is it visible?
Yes, ma'am, it's visible.
So here, as I have mentioned, that it is 2. Okay, so in the first one, you can see Department of Microbiology. I've just taken random example. Okay,
Sorry for the interruption, ma'am. Can you please move the slide? It's come back to the same slide.
Slide is move, not move.
Yes,
no?
now it's visible. Yeah.
Correct.
So, Department of Microbiology, Arts, Commerce and Science College, Xavier College, Mumbai, Maharashtra, India. Okay, whereas second, which is there, it says that the person is from different institutes. So, it is Department of Microbiology, Dr. D.Y. Patil College, Pimpri, Pune, Maharashtra, India. So, this is how you have to write the affiliation. Okay, then comes, so you have chosen the corresponding author. Who is the corresponding author? That is also chosen. Okay, then comes the email address of the corresponding author. So as I already mentioned, the corresponding author is responsible with the communication of the research articles, and hence you need to give the email address after the affiliation. Okay, after the affiliation, you need to give the email address of the corresponding author. So corresponding author, if not star, you can also denote it as envelope, which is shown there. Okay, a small envelope. So either of it you can show as the symbol for the corresponding author. Okay, that denotes that among this group, this is the person who is the corresponding author, because it should be identified. Okay, so now, research article, whenever you want to communicate any publication, okay, because just doing the work is not important. You also need to be communicate your research work. Otherwise, whatever the work that is there, it will not be known to the people. Okay, so whatever the work is there, and it should also be important to the society. So there are different ways of how you can communicate your research work, and they are in the form of research articles, short communication, or you have the review articles, books, book chapters, patents, some of the scientific reports, okay, or some of the magazines also. So these are the ways you can say by which a person can communicate his research work. Now, as I said, we are following the IMRAD pattern. I stands for introduction, M stands for materials and methods, which we call, R stands for result, A stands for and D is for discussion. So this is the pattern, okay? After you are done with things, so title, then you are having the authors, okay, or a single author can be there. Then you have the affiliation. After affiliation, you have to select, there is a mutual understanding between the authors, okay, who wants to be the corresponding author. So that there has to be a mutual understanding. And then after that, the email address of the corresponding author. So once it is done, then the next step is the abstract. Comes the abstract, okay. So in abstract, what are the points that need to be covered, okay? So from your title, you have to see how to write the abstract. Okay, so in the abstract, these following are the points which you need to cover. What is the problem that you are going to tackle? Okay, so how the problem is there and how you are going to tackle it. Why is it necessary to tackle such problem? What is the solution? Solution means what method you are going to use so as to tackle that problem. So it is the solution. Solution means the method, approach, or the method. It has to be in very short. Okay. Then what are the results? The main results have to be highlighted. Okay. The significant, not all the points, but the main significant results, they need to be taken into consideration in the abstract. Okay. So whatever the result, highly significant ones, you need to mention. What is the conclusion and what is the significance of the work or the application which is there. Okay, so you have to conclude it properly. And what is the significance, like whether it is important to the society, how is it important, or whether it is important in case of agriculture, then how is it important, how will it be important to the farmers. Okay, so these things will come under the significance. Then after that you have the keywords. Now keywords, after the abstract, immediately below is the keywords that you need to mention. 4 to 5 keywords are generally asked. Now see, the keywords are very important things that you need to consider. Like, what is, so from the title only you cannot take the keywords. Okay, so whatever the words they have come in the title, you are not supposed to take them in the keywords. Keywords have to be very different. Okay, so like which is related to your work. So if I take for example waste management, okay, so my I can take the keywords as environment, or I can take the keyword as economical, or I can take the keyword as eco-friendly. So this is what it is. Okay, or I can take the keyword as uh, incineration. So these are the following important keywords you need to select. Okay, 4 to 5 keywords are generally asked. In some cases, the journals, they ask about 6 keywords. Okay, so that will depend. Then after this is done is the introduction. Now introduction, when you are writing the introduction, it says that it is the important aspect of your research topic along with recent citations that should be taken into introduction. Now before writing the introduction, you just have to read what is the title. Okay, what is the title of your topic. So based, taking into consideration that title, you have to cover all the aspects of your title and write down the introduction. Okay, so this is how you have to, you have to expand the things in the introduction, each aspect which is there in your title, that you need to expand it. Okay, so along with the citations, okay, I have mentioned the example. What is the citation? Like I have taken it, Gunjal et al. 2018. 2018 stands for year. Gunjal is the surname, and ETAL is that, along with me some other people are there, co-authors, which we call it as. Okay, if there are more than 2 authors, then you need to mention it all. Okay, if there are 2, then you can say only, like for example Gunjal and Sharma, then you mention the year. Okay, comma, right comma, and then it has to be followed by the year. But if there are more than 2, then you have to write ital, ETAL, and that has to be in italics. Okay, which you will find the option in word format. Okay, italics. So this ital has to be written in the italics. So introduction is like, you, as I said, you just read the title. Okay, and that you go on expanding. Now whatever, when you are writing the introduction, there is a tendency that you copy the things from different research paper. See, now we are not supposed to copy the things. Okay, because that counts for the plagiarism, and plagiarism is very strict. So we, because once you write the research paper, it goes through a software to see that the things are not copied. Okay, and that is called as the checking for the plagiarism, and there is a software to check the plagiarism. Okay, as we move ahead, I will go, tell you about it. But nothing is to be copied, okay, from any of the things, because now earlier it was allowed 10% plagiarism, but now even 10% is not allowed. Okay, so copying is strictly prohibited. Okay, so like there is a tendency, most of the people they don't know, so they will just copy the things and they will make the introduction from different research papers. They will, so you have to write it in your own words. Okay, so please keep this in mind before you write the introduction. And you have to, like, you can read the research paper. So once you read the research paper which is related to your aspects, different research paper, so you get the idea what is being there, what are people doing, what work is carried out, how much is the work that is done, and based on these things you get the general idea. Okay, so taking into all that consideration, you have to write the introduction in your own words which is there. Okay, and for that you have to read all the research paper, recent ones which is related to your work. Then comes M. This stands for the material and methods. Okay, so mention the methods, protocols, or assay which is followed on your research work in short. Okay, if a method, protocol, or assay is followed from a particular research paper, then you have to cite that particular research paper. Okay, citation, as I said earlier, also how you have to do it. Okay, if it is your own method that you have developed, then detailed description is required. Okay, then it has to be done in detail. Okay, and a method, whatever it is there, you have to write it in paragraph form. Okay, it is not to be written in a flow sheet. If it is a protocol, then also you have to write it, or assay, it has to be given in paragraph. Okay, so introduction also it has to be given in paragraphs. Okay, so other materials and methods also, if in case there is a schematic representation, then you can make the pictures, images, and you can make the flow sheets. Okay, so that is how it is. If the method is new, if it is developed by your own, then you describe it fully. Okay, in materials and methods, so you have to even mention the model and make of the instrument or the equipment. Model and make, like, of the sophisticated instruments. Okay, so you have to give their name and from which country it is. Okay, make, that we call it as make and a model number. Okay, and from which country it is. Now, instruments, sophisticated instruments, for them only you have to give the model and a make. Such as sophisticated instruments, you have UV visible spectrophotometer, you have phase contrast microscope, then you have scanning electron microscope, or if you have done transmission electron microscope, polymerase chain reaction machine. So all these are the sophisticated instruments. In that case, you have to give the model and make. If it is Fourier transfer infrared, okay, FTIR, which we call it as, or atomic absorption spectrophotometer for the study of metals. In that case, you need to give the name, make, model of such instruments. Now, these are the sophisticated instruments. So, model and make for weighing balance, pH meter. Okay, for that it is not required, or for the water bath it is not required. Okay, sophisticated instruments means the sophisticated instruments which I mentioned earlier. Okay, so they will come under the class of sophisticated instruments. For balance, for pH meter, it is not required. Okay, or for water bath it is not required. For oven also it is not required. Okay, so materials and methods, it has to be described properly and in details. And as I said, if it is new method, then complete description has to be given so that third person is able to understand what is there in the related aspect of the paper. Okay, so that is how it is there. Then comes results, which is R. So you need to mention the results. Okay, one by one mention the results that is there. Okay, of your research work in a proper way. It is necessary to give the accession number. Like for microbiology, if you have isolated some new organism, then it is expected that you give the accession number. You have the complete genus and the species, and you submit that whatever it is there, submit it to the database, those database, they will give you the accession number. And that accession number has to come in the research paper, okay, which because reviewers they expect any the results, materials methods one by one you have to take, okay, you cannot pile up or you cannot mix it. So one by one objective you have to take, and for each objective as you are proceeding, you have to write the materials and methods. Similar way, for each objective, whatever the results you have got, you have to mention it in a proper way. Now results, they can be mentioned either in tabular or graphical form, okay, but both ways is not like, if you are mentioning the result in graphs, okay, then the any same result can be represented in table form. That so either of it you can use it, okay, not both, because that will become the repetition. So either you mention it in the graph form or you can mention it in the table form, okay. So that is what it is. As I mentioned, both ways, if your data can be presented, it is well and good, but only one of it you have to choose, okay. That depends on the choice of the author, but both you cannot select, because the thing is that it becomes the repetition. So repetition of the data is not allowed in the paper, okay, because that becomes a poor reflection, okay. So that is to be avoided. If the data is very vast, you can compress it, okay. You can compress it so that it becomes very easy. You can see to it that if you apply some statistics, that will be very good. Any tables, figures, they should have proper headings and captions, which is very, very important. If you are making the figures, you have to like graphs, okay, whether it is line graph, whether it is bar graph, whether it is histogram, any type of it, whether it is pie diagram, you have to give the axis, okay. You have to label the axis. Even the title to the figures have to be given, title to the tables have to be given. Without that, it is not a good quality, okay. So that is what it is. Any short form in the table, if you are using in the short form in the table, it can be used, okay. But those short forms which you have used in the table, that you mention it just below the table, immediately below the table as the footnote, okay. So that the reader, whoever is reading your paper, because the third person, he might be new, he or she might be new, so they might not immediately understand, okay. Because whatever work it is there now, it is, we have to see it in terms of the common person, okay. Because a common person should also understand, okay, all the things that are being done, okay, and how useful it is. So even if you are using the short form in the table, you can use it, but just mention it immediately below the table in the form like full form, okay, in the footnote, okay. The data presented in the tabular form should not be repeated in the graphical form, okay. By mistake I have written it that it can be repeated, but any one of it you have to use it. Now this is the example, okay, of the table. So I have mentioned the title, okay. It is the title, removal of zinc and lead. I've just randomly taken one of it, okay. One example, zinc and lead by dead biomass of Aspergillus and penicillium, okay. So this is how, wherever required, you have to mention the units, okay, because that is also very important. Units wherever required, it is not that units might be required everywhere, but wherever required, you give the proper units also, okay. Any plus or minus, here it indicates 0 0.02, 0 0.00, it 0 0.01, okay. This indicates the standard deviation, okay. So that the third person is aware, he or she knows what is the standard deviation. It indicates, or the reviewer also comes to know that this person or these authors, they are aware and they know that whatever it is there, the data has to be in replicates, okay. It ensures that your data is authenticate or your data is correct, okay. and the experiment, whatever it is there, it is repeated 3 times or more than 3 times, okay? Because whenever you are representing the data in tabular form, if it is assay, okay, if it is a standard graph, or if it is some enzyme production, the quantitative values, okay, so here it is removal of metals, as you can see the example. It says that the data is repeated more than 3 times, and hence why is it necessary? Because it gives the authenticate of the data. It is 100% correct, okay? That is why you need to repeat. Now, not for all cases you have to show the replicates, okay? Like, for example, microbiology, if I talk about morphology of the colony characters. Now, there you cannot show anything in replicates, okay? So, such table which is there in that case, the replicate, like standard deviation is not required, okay? Morphological characteristics of the colony, where everything it is there. So in that case, the standard deviation will not come. Or in case of oxidase catalase, so such are the things, okay? Sugar fermentation, in that case, you cannot expect the standard deviation. Standard deviation is the thing where the quantitative values will come, and for that quantitative values, see, because here it is the quantitative value, so just doing once it is not suffice, okay? And that does not indicate the good quality of a research. So where the quantitative values are coming into picture, if in case, then you need to repeat the data and you need to show the authenticity, and hence there is the necessary that you do the replicates, okay? So, and you mentioned the standard deviation or the standard error, okay? So that is why it is necessary. So graphical representation, coming to the graphical representation, the data in graphical form can be presented by, you can see this in Excel, okay? There are different options: by line, pie diagram, histogram, bar diagram, scatter diagram, okay? So these are the different ways by which you can present your data in the graph form, okay? And these options you will find in the Excel. You have PowerPoint, you have Word, and the third is the Excel. Now this is, I have mentioned, the data needs to be reproducible and reliable. Hence, okay, if the data is very large, okay, if your data involves some survey, then you can compress it. And this is how the line diagram can be. You have to label your x-axis, y-axis, you have to show the legends, okay, and you have to give the title to your figure. Now when you are giving the title to your figure, the title has to be below the figure, okay, as you can see here, okay? Figure the biomass in milligram of microorganisms A and B at different time interval in nutrient growth, okay? But for tables, the title has to be above the table, okay? In research paper, you must have, like, some of you may be knowing, okay? The title of the figure is below the figure, but for table, it is above the table, the title that is there, okay? In case of, like, you can say, figures, you can even show the error bars, okay? So that error bars, they say that it is authenticate. Here I have not shown, okay, but you can show the error bars, okay? And this is a scatter plot. I have just shown the different form. Earlier was a line graph. This is a scatter plot of a same, okay? Anyway, you have to remember that you have x-axis properly. Your line should pass through origin. If it is a standard graph, you have to label the x-axis, y-axis, and for a scatter plot, you can show the straight line equation and the r value, okay? Now in results section, some of the things, they are important. As a microbiologist, as I mentioned, accession number is very important. And once you do, like, you have studied the morphological characteristics, biochemical, okay, biochemical, like oxidase catalase, citrate utilization, Then you will go to the Burgess manual and you will do the sugar fermentation test. So these are some of the basics, okay? But it doesn't, like, you will just, then you will compare it with the Burgess manual and you will get the genus. But this is not suffice. Even the species is required, okay? Because like for a very good paper, you require both the genus and a species. And for that, you need to reach to the molecular level, authenticate level, and that is by doing the PCR. Before that, you need to isolate the genomic DNA. Once you isolate the genomic DNA, then you can go for the PCR. You can amplify the product, which is called as the polymerase chain reaction. Once you amplify it, whatever the sequence you get, okay, that you have to submit it to the database, such as the NCBI, National Center for Biotechnology Information. Now that sequence, once you submit, automatically those database centers, they will give you the accession number, and that accession number is very important, okay, which has to come into your research paper for publication. In case of experiment involving animal models, okay, for testing, some of the experiments you might require the use of animals, okay. So in that case, ethical permission has to be taken, and that permission has to be joined, okay, when you are submitting the research paper, okay. There is the option when you are submitting the research paper, okay, wherever required, if you require, if okay, ethical permission, you have to take it and you have to attach it, okay, when you're submitting the paper. Like for example, if in case, okay, not all of it might require use of animals, okay, or use of some toxic drugs, so in such case, or lethal drugs, in such case, or some carcinogenic, if it involves some carcinogenic compounds, if you are working with, then yeah, for that disposal you might require the ethical permission. So you have to take the ethical permission, and they might give you some certificate, okay, to carry out the work, that you have to put it when you are submitting your research paper, so that the editor is also aware of the things, that these authors, they are quite they know what is ethics in research, how to take the ethical permission. So all these things they are seen, and based on that, your paper by the editor, all these things, is taken into consideration by the editor. You'll see, if the editor feels, then only your paper goes to the reviewers, okay. Otherwise, there itself it is rejected by the editor, okay. If the editor feels that it is not worth to send it to the reviewers, it is rejected there itself by the publisher, okay. Then the experiments involving carcinogenic or toxic compounds, the disposal methods, it should be mentioned in the research paper. Statistical analysis is also very necessary. Like for example, in case of pot experiment, field experiments, randomized block designs, okay. So you need to see that you apply the statistical analysis wherever it is important, because statistical data is also carrying the weightage, okay. So you can apply the test, t-test, chi-square test, f-test, ANOVA, which is the analysis of variance, okay. So you have to see and you have to report, because if it is pot experiment, field experiment, if your work involves such type of the experiment, you need to keep the sample under control. You have to see how much significant difference is there between the sample and the control. How good is your sample when you compare it with the control, okay. For that is called as the significant difference. I need to see how good it is. It is good, okay, it might be good, or it might be at par. At par, that means it is at level. Sample and control can be also, okay, at par. So we say that it is at almost the same, okay. Or the second option, or the second can be that test is more good, okay, it is highly good, but how good? So for that you have to see the difference, and in that you can apply the different tests which is mentioned here, okay. T, chi-square, F test, and analysis of variance. Then coming to the discussion, D, compare the results. Okay, so you have written the results. Now in result section, one has to keep in mind that just drawing the MSc students like we are observing, or even the faculty members, most of the faculty members, they don't know, or even the MSc students, just drawing the figures or just giving the tables is not suffice. You have to elaborate it. Okay, so before you put the tables or before you put the figures, elaborate them, mention them, what is this. Okay, because many of the people, they think just by putting the tables the work is done. It is not the thing. Okay, just looking at the figures, tables, uh, a third person might not be able to understand, so you have to highlight. Okay, highlight the text. You mention first what it is. Okay, what is important in your result. So each, one by one, you have to take it as it is coming. Okay, my objectives. So one by one, you have to describe the results. Okay, and you have to like expand and explain what is your figure telling, what is your table telling. Okay, how which is the highest value, which is the lowest value. Okay, it can be, but you have to mention it. So now in discussion, compare the results obtained with similar aspect of your work done by other researchers. Okay, check the novelty of your work. Okay, now in discussion also, you have to take the citations because you are referring to different research paper. So citations will be in introduction, discussion, and in methodology. Methodology, if you are referring to some method or protocol, then you have to cite it from some research paper. There the citation will be there. Okay, and in discussion also it will be there because you are comparing your results with similar aspect. Okay, it will not be exactly similar, but somewhat similarity you have to see, and based on that work. Okay, your work, and different related to your aspect, you have to take into consideration, and you have to mention how novel is your work, or it is at par, what values you have got, whether it is really very high. Okay, with the work done by other people, or whether you have got some, you have isolated some new organisms. So, and how is it important with respect to the industry, how applicable it is. Okay, or is it producing some good enzymes, so commercialization is there, or if it is related to the agriculture aspect, how important, how novel it is to the agricultural people or to the farmers that is there. All that has to come into aspect. Okay, how novel is your work, or whether it is the same, because there has to be some novelty. Novelty means something new. Okay, routine or the same work that is done, it is not of any use. Okay, because such type of work, then it is already people are known. Okay, so there has to be some novel, and that you have to catch, and you have to accordingly select the topic. Okay, doing the same work which is done earlier is not of any importance. Okay, because such type of work is already done by some people. So if you are doing the same similar type of work, it is just the repetition. Okay, so it does not carry any novelty. So you have to see some novel aspect. You have to accordingly select the title of your work. Accordingly, you have to work. Accordingly, you have to see the significance of your work. Okay, now depending on the format of the journal, the result and discussion may be separate or it might be combined. Now there are variety of the journals. Okay, some of them, they say that result and discussion can be combined in one, or some of them, they say that result section has to be separate and the discussion section has to be separate. So these things has to be followed. Okay, so such is the thing. Then your section is the conclusion section. Okay, so this is what it is. Okay, after the discussion, So you have the conclusion section. Okay. Now in conclusion section, this also has to be precise. Okay. It does not has to be a long story. Okay. So you have to include the significance of the research work. And many people even they don't know like conclusion section. So conclusion is output of your work. What is the outcome? What is in short, it is the outcome. Okay, so it is not the discussion. So many people combine or they will take a conclusion as the discussion. Okay, so that is not the thing. Conclusion is the ultimate output, and what is the significance of your work and its application. Okay, how will it be applicable? Then comes the acknowledgement section. Now, acknowledgement is in some cases, okay, it might not be applicable to all, but wherever it is applicable, you have to include this acknowledgement section. Acknowledgement means thanks. So, like for many projects or for many research work, there are fundings available. Okay, you have the government fundings such as the Department of Biotechnology, Department of Science and Technology, Council of Scientific and Industrial Research, okay, Indian Institute of Science. So all this they provide the funding for your research work, okay, or for your projects which is there. If in case you have funding from such government sections, okay, or from such government institutes, then you have to acknowledge them, okay, at the end, in the sense that you have to thank them and say that they have provided the funding for your research work. This has to come in the acknowledgement section. And you have to say, okay, I'm thankful, or we are thankful to so and so for providing us the funding for our research work, so and so. So this is one or two sentences which is very necessary, although it is one or two sentences, but these are the government institutes or the government people which are providing you the funding. And it is also like undertaken that if people or the students or the people they publish paper, okay, and they DST, if they provide the funding, they undertake that if you are publishing the paper, you have to acknowledge us. So this is called as the acknowledgement. The other way of acknowledgement is some people they are there who help you in your research work. Maybe they might help you in statistics, okay, or they might help you by providing you some culture, or they might help you in some microscope work, or they might help you in some your pot experiment, or they might help you in any other way, okay. Then the person whoever is there, he or she has to be acknowledged. So this is called as the acknowledgement section, okay. Then is the references. Now all the citations, as I mentioned, whatever the citations they are coming, okay, in your discussion, introduction or methodology, it must be mentioned in the references. References is nothing but you are expanding the citations, okay. So that is called as the references section, and all the citations they have to be expanded and they have to be covered in the references. Now, each journal which are there, they are following their own style of references. So, whatever journal you want to communicate your research paper, you have to see their guidelines, okay. Each journal they are having their website, so they give the guidelines to the authors how the references style has to be, okay. So you have to follow exactly that style. So there is one where the variation can be there because there are many journals, each of them they are following their own style. Or the other thing is, as I mentioned, some of the journals they say they combine the results and discussion. Some of them they say you keep the results and discussion sections separately, okay. Some of the journals, so here is one of the variation. Some of the journals, they say that font size of the whole text has to be 10. Some of them, they say that the font size has to be 12. So here also the variation is there. Okay. Some of the journals, they say that you give even the graphical abstract. So not only the abstract which I mentioned earlier, but some of the journals, they say that you give the graphical abstract also. What do you understand by graphical abstract is that all the work which is there, you have to show it in the form of a picture. Okay, so the whole section which is there, you have to combine it all and show it in the form of graph. Okay, the whole summary, right, starting from the whole, you have to, so that some of the journals, okay, so they might ask for graphical abstract. So here is the variation. Not all of them might ask, but here also you will find the variation. Some of the journals, they say that you keep the font as Times New Roman. Mostly it is Times New Roman, but some of them, they say that you keep it as Caliber. So here also the variation is there. Mostly it is Times New Roman, most of the journals, they say. Some of the journals, they say that you mention the email ID of all the authors. Okay, some of the journals. Some of the journals, they say that you give the email ID of only the corresponding author. Okay, so there also you will have the variation. So this is how it is there. Some of the journals, they say that you give the line number. Okay, right, starting from the beginning till the end. So your text has to be given the line numbering. Some of the journals, they do not ask for line numbering. This option of line numbering you will find in Word. Okay, so there is a continuous line numbering, so you have to have it accordingly. So here are some of the style, I have just mentioned it. Okay, now some of the points which are tricky and which will help you of good quality research paper is data has to be novel, it has to be reproducible and authenticate. Always remember this. And whenever if the third person repeats your work, authenticate, reproducible means that whoever the person, if he or she has repeated the same work, he or she should also get the same result. This is called as reproducible and authenticate. If it is not reproducible and authenticate, there is some problem. Okay, that means it is not 100% true. Data has to be significant, having large scale applications. So now mostly we are like, even our government says that whatever it is there, your product, or you are developing some new technology or some process, it has to have some application. Okay, it has to have application in the society, or it has to have some application in the industry, or it has to have some application in the medical field, or it has to have some application in the agriculture or to the farmers. So such is the application, variety of applications. Okay, just because now people should also get the benefit of the thing that is there, because you are, see, research, the word itself says that you are finding some solution to the problem. Okay, so it has to be the way it is. Plagiarism, now as I mentioned, this is very important. So once your draft is ready, it is once you submit it, the editor may themselves check. It has, you yourself can check for the plagiarism. Of course, you have to do it. You can, I will not say, but you yourself check. Ani, once it is checked, there is a software which is called as Turnitin. You get a report. I mentioned that it has to be less than 10%, but actually, even 1% plagiarism is not allowed. Okay, simple is that copying of anything is not allowed. Editor may also check the plagiarism. Okay, if they find there is a high plagiarism, they will reject it there and there itself. If the editor does not check, even the reviewers can check the plagiarism, because there are options. Okay, so even the reviewer, the reviewer will directly, if the reviewer comes to know that it is highly plagiarism, there itself they can reject the paper and inform the editor. Okay, so using this software Turnitin, you can check the plagiarism, and you have to see to it that everything is good. Okay, that ensures the good quality of the work, because now plagiarism, there are many cases, and people who have done the plagiarism, the publishers, they have prohibited from taking their papers. Okay, so even the publishers, they will not accept such persons research papers any further. Okay, and the good publishers are Springer, Elsevier, Wiley, Taylor and Francis. Okay, these are the very good publishers, and you should refer their research papers, what people are doing, how they have written the paper. Okay, how the images they are drawing it, how wonderful the images are, how the table, the make of the tables is. Okay, how the art is, because some of the images, scanning electron microscope images, transmission electron microscope images, okay, they are very, very beautiful. So nature, of course, your work also, nature journal, it is the topmost journal, but your work also has to be of such quality. Okay, so this is all about how a good research paper is, and thank you for your patience. Yes, if there
Thank
are
you.
questions, I will be happy to have them.
Yeah. The session is open for question and answers. If anyone wants to raise any question, you can unmute yourself. Ma'am, we have few questions. The question is from Prakash sir. How statistical analysis is important for biological samples?
See, if your work involves pot experiment, field experiments, there you can apply these statistics. But with respect to the if it is just dealing with the simple, like if you are not having any pot experiments or field experiments, then you did not apply the statistics. Okay, just simple use of organisms is there, and it's and for example, if it is enzyme production and such things, there the statistics is not important. Yes.
Next question. To what extent plagiarism is permitted for STI manuscripts and PhD thesis?
I already mentioned no. No
Yeah,
plagiarism.
ma'am. Yes. Ma'am, one question from my end. You have mentioned one point of novelty in your work.
Yeah.
So my question is, like, now many of the students are behind PhD and they are looking into getting their PhDs, and to what extent should be your work novel? Like, if you can define it in number of percentage, like, or to what extent the novelty can be? Because every time it's not very easy to find out novelty in work, because there are many things or many work that is going on. Just like for enzymology, lot of enzymes are worked on and lot of work is going on still. So if any research scholar wants to work on enzyme, and if he or she is asked for novelty, to what extent can we expect the novelty in every research work?
See, yes, novelty has to be there, and I'm like you can have the novelty, so like for example, novelty is, so for that, you see, what you have to do is, suppose there is a topic in your mind on which you want to work. Okay, there
Okay.
has to be some topic, so you just put it on the Google. Okay, different keywords you have to put. Okay, just so related to that, and you have to see what is the work, such work which is, whether it is done, or what type of it is done. So, Ani, if you are talking about the enzymes, so you have to see which there is any different enzyme. Okay, what application it can have. So you, like for example, you can screen the organisms and see if they are producing some different enzyme. You have to see for its production, and then you have to study for its novelty. Now the most novelty is, you cannot study the enzyme amylase. Okay, because that is studied in lot. So that we cannot take amylase, for example, okay, because a lot of work is done. At the most, what you can do is you can study some aspect related to some other enzyme. Like, for example, there are many other enzymes. If I take into consideration carboxylase, okay, or some 1-amino cyclopropane decarboxylate. So these are the enzymes on which work is not done. ACC deaminase, PGPR, these are producing the ACC deaminase. So on this, like if you see this one enzyme which is there, I gave you just now the enzyme. So this is one of the enzyme on which work is not done. So novelty, you have to find the novelty. Okay.
Okay, ma'am. One more question is, which are other publications
One-
other than you
one-
have mentioned?
One thing,
Okay.
hello.
Yes, sir. Yes, sir. You're audible.
You are saying regarding the novelty. Okay.
Yes,
And
sir.
the example it is given of the enzymes.
Yes,
But
sir.
what happen? Enzyme may be the same, but for many more time its source may differ. It's a
Yes,
novel
sir.
source. It's a novel source. Its properties may be different.
That's what, sir.
Yeah. So if it is there, then it's a novel. Enzyme may be the same, but other things may be different, which are not
Yeah, so
included earlier.
because nowadays, whenever you apply for any PhD, you can't every day find out a new and new topic, because now it's a long time where PhDs are getting and work is done. So every time if you ask for 100% novelty, it's really not possible in
No, not 100.
certain fields.
Not 100%, but these are the novelties. Suppose
Sure,
all
sir. Sure, sir.
amylases, they are having some optimum temperature.
Yes, sir.
But your enzyme, the same enzyme activity is same, but it has
Huh.
got a optimal temperature. It is a very different one.
Different temperature. Yes.
And then the
Or you
properties
can,
also go on changing.
Yes, sir.
So,
you
so
can
that,
even study some different applications,
yeah,
Yes, sir. Yes, ma'am.
application,
Yes.
some novel application.
or it may be produced by some other organism, which is not
Okay.
researched yet.
Yes, sir. Yes, sir. Thank you, sir. Ma'am, the other question is, which are the other publications other than you have mentioned?
You have SAGE, SAGE, then you have World Resources Association. So these are good.
Okay. One more question is, there are certain research papers where materials and methodology are written after discussion instead of after introduction. So which order should be followed?
No, that is wrong. We are following MRAD pattern only. So
Yeah,
after
yeah, ma'am.
introduction, it has to be there. Yes.
Okay. One more question. Yes, sir.
Question, madam, correct. IMRAD methodology should be followed for research article. That's why I say.
Ma'am, one more last question.
Yes.
Is it necessary to subscribe the journal for publishing in it?
No, not required. Not necessary. Not at all.
Okay, ma'am. Any questions? Any more questions from participants end? Is there any alternative for Turnitin for plagiarism checking?
No, Turnitin is the best.
Yeah.
People are using, yeah.
Yes, ma'am. Apart from that, even according to my knowledge, there are few, but what you have said, Turnitin is the best amongst all of them.
Yeah. That is authenticate.
Yes, ma'am. Yes. I guess the questions are done. So it was really a very detailed session where you have really explained each and every aspect of how to write a research paper, what are the authors. I would like to thank today's speaker, Dr. Aparna ma'am, for giving her valuable time and letting us understand how to write a research paper. So I would like to show my gratitude towards Dr. Aparna ma'am on behalf of Microbiologist Society. Thank you, ma'am.
Thank you. Yes. Welcome.
Thank you, madam.
Yeah, I would also like to thank all the participants who have joined for the session today. And I guess we are very happy today. We have the the highest participants, almost 67 participants we had. So I would also like to thank all the participants who have been actively present for the session and made it a very nice event. And last but not the least, I would like to thank Dr. Deshmukh sir, who always gives us the opportunity to conduct sessions like this and explore the unknown areas in life sciences and research related to life sciences. Thank you. Thank you, ma'am.
Thank you.
Yeah,
It was
yeah.
my pleasure
Thank
and
you.
very nice to share the things.
Yes,
because
ma'am.
that
Yes.
is very important. And
Ma'am, it will be helpful if you can share the PPT so that we can
Yes.
circulate it in the group.
yes, I will send it to you so that you can.
Sure, ma'am. Sure. I'll share it in the groups.
Yeah.
Yeah. Thank you, ma'am. Thank you for your time.
Yes.
Thank
Okay.
you. Yeah.